Thank you, Bethany. Thanks for hanging in there, everyone. Good afternoon. It's my pleasure to present BioLife Solutions. I think there's one more session after me, so I'll appreciate your attention. Bob Pretty suggested I pass out bottles of Cryostore, but I think at this time of the day, I need to give it something a little stronger. Please note our safe harbor statement here. So BioLife Solutions is based in Seattle and Bothell. Our mission is to become the leading provider of biopreservation tools for cells, tissues, and organs. How do we do that? We want to help you folks facilitate research and commercialization of biologic-based medicine. Specifically how? By improving yield and functional recovery of not only source material, but manufactured cell products during manufacturing, distribution, and patient administration. Here's a shot of one of the two buildings that we rent space in Seattle. We have 30,000 square feet under lease, 2 independent GMP suites. We can make 125,000 liters of product annually with no CapEx investments required. BioLife has been public as a penny stock for many years, but in the first quarter of 2014, uplisted to the NASDAQ, where we trade right now. We have just a little under 50 team members, and we expect to break even sometime next year. Pardon me. Some growth drivers. So we've been enjoying 30% annual revenue growth, serving drug discovery, biobanking, and the RegenMed space. To date this year, we've gained over 150 new customers. We're really proud of the fact that with your help and your support, Cryostore and Hypertherm Assault are now embedded in over 230 customer validation projects and clinical trials. I'll have some more detail about that a little bit later. And you might know that we have this disruptive biologistics cold chain management SaaS application and the Evo SmartShipper that we've been supplying to the market. I'll talk more about that later as well. The company's received a number of awards for culture, and it's a great place to work, and we have a very dedicated team. We are very focused on supporting customers, and we really get it that quality is really what it's all about. And we're doing what we're doing because the same reason you are, this is all about patients, so we take that very seriously. My executive management team very successful executing for growth in a number of businesses. I won't read this right now for the sake of brevity. So let's talk a little bit about our 3 key markets. So within biobanking, we're selling our products to cord blood banks, to adult stem cell banks, tissue banks, biorepositories, and a number of hair transplant physicians who use Hypertherm Assault to to preserve this explanted strip of tissue and the dissected grafts ex vivo before they're transplanted back into the scalp of the patient. Within drug discovery, we're selling directly to big pharma, to a number of cell suppliers, tox labs, and personalized medicine labs. And to us, most importantly, of most strategic interest to us is this space, the cell therapy or RegenMed space, where we're selling to commercial companies, hospital-based stem cell transplant labs, and the university-based upstream development labs where a lot of this technology emanates from. So a little bit more specific about RegenMed is the way we see it. So this is ARM data from the Q2 data report. Maury shared some updated numbers earlier this morning, so really great level of investment. Why do we have a company? This is all about preservation of biological material. So once we take cells or tissues outside the body, we can have a number of applications that we can use these for: solid organ transplant, tissue biopsies, tumor analysis, apheresis and leukapheresis collections, bone marrow transplants, so on and so forth. The challenge is once we do this, this biological material starts to degrade. It's literally a race against time. So we use hyperthermic storage, either in a refrigerator or a frozen mode, to reduce metabolism, to literally buy some time so we can push this source material and these final manufactured products around from point A to point B. We see a yield as really important, and not just survival, but functional recovery of cells so they can come in from the cold and do what they're supposed to. And this is, to our view, this is a continuum. So if we can help our customers improve yield of source material, we can make more doses out of the same given starting material. If we can improve yield, the final manufactured dose, we can achieve the desired therapeutic effect with fewer cells. And many customers, in fact, folks in the room here today, use our products both on the front end and the back end with the final manufactured product. And yield improvements can be monetized, which is really important, obviously, as we're looking to commercialize these life-saving therapies. So we have a vein-to-vein engagement. I'll talk more about the Evo SmartShipper in a moment, but we have really 2 opportunities on the front side, from the patient, from the bedside to the cell factory, pushing the source material, bathing it in an appropriate preservative so the cells or tissue can stay alive, putting it in a shipping container that can maintain the temperature. Once the manufacturer's cell product is completed, pushing it back to the clinic again in an appropriate container, in an appropriate media. In many of the customer clinical trials, our products are used as an excipient, so the actual are the diluent or the injected, if you will, the infusate to deliver these cells to the patient with no wash. Again, we're going to start with media and then talk about Evo and biologistics in a minute. So you may understand or know the brand names Cryostore and Hypertherm Assault. Cryostore is a clinical grade freeze media, serum-free, protein-free, animal origin-free, multi-compendial components that are used, manufactured per CGMP in our facility in Bothell. So Cryostore has been shown to improve yield in a number of cell and tissue types. And we've got a lot of evidence on our website that I'll point you to in a moment. So Cryostore, way back in the day when it was formulated in the companion Hypertherm Assault, are the result of some experiments that some scientists did to unlock some of the mysteries, if you will, of molecular-based cell stress that is induced when you make cells cold. So when you subject cells or tissues to hypothermia, it's not just the mechanical damage, it's what's going on a molecular basis. And these scientists at the time did a good job to understand what sort of components or ingredients they would add to the media to mitigate some of these pathways. And it works. Hypertherm Assault is intended for 2 to 8C storage and shipping of cells and tissues, again, serum-free, protein-free, animal origin-free. And we have customers using Hypertherm Assault to ship starting material and final products, not frozen, but in a fresh 2 to 8C mode where they're not thawed. They don't need to be thawed. They're simply sprayed off and then infused. A couple of fluorescent micrographs here. You can see cells preserved in Hypertherm Assault on the right. These are human MSCs isolated from bone marrow and preserved here in a traditional home brew of culture media with some serum. 5 days in the cold, then returned to culture condition, then assayed to see what the true functional recovery is. And you can see a nice confluent monolayer of cells here with mitochondria, the nucleus, and the cytoskeleton going on there compared to this. This is why we have customers. The customers who've adopted or embedded our products in their trials obviously put the products through a number of comparative experiments, and these are the results that they see. A little bit more about the clinical trial base right now. This is the franchise that we built. So today, 230 customer validations in clinical trials, about half in blood cancers and solid tumors, but really all the major disease states targeted. And we think we can monetize each one of these indications should the customer achieve regulatory approvals and commence commercial manufacturing at $500,000 to $2 million a year in annual revenue. Obviously, they're not all going to make it. There'll be a lot of attrition. Nevertheless, we built a wonderful platform and a franchise going forward here. And we look to really the first couple quarters of next year for some exciting news from some of our early customers. This is another cut of the data that shows by clinical indication here and by phase of development how these clinical trials our customers are running stack out. So you can see 21 in phase 3, almost 100 in phase 2. And again, with most all the major disease states targeted here. I mentioned evidence on our website. So today on the evidence page, you can find over 200 articles, abstracts, and posters if you're interested, covering a broad array of cell and tissue types. What is the size of the demand from preservation media? It's almost $1 billion here, driven from RegenMed. Now I'm going to shift gears and talk a little bit about biologistics. So this is a cold chain management SaaS application that's really embedded with or bound to a new innovative smart shipping container for biologics. So I pulled some data from a couple of traditional pharma research reports, and really this is the comparison and contrast I'd like to make for you. You can see in traditional pharma, I mean, this is a serious number of shipments that require temperature-sensitive logistics. Call it 100 million, if you will. Annually, $12 billion is spent on these cold chain logistics, with $9 billion for transportation and $3 billion for specialized containers, data loggers, so on and so forth. Now let's talk about the bad news. So 15 to 35 billion is spent annually replacing products that were lost due to temperature excursions. So that's about 6% of revenue from cold chain pharma being spent on logistics. Yet if you spread that out over the total number of doses that are shipped, it's really a de minimis spend. Now I'll contrast that to cell therapy with these personalized medicine. Temp sensitive, in many cases, the patient's last hope. They might not be alive for a repeat infusion or apheresis, if you will. So fully burdened, we know some of these cost estimates, maybe they range from $30,000 to $50,000, and perhaps reimbursement might range from $100,000 to $500,000 or more. And this is really critical, the risk of the dose not showing up in time or being exposed to a temperature excursion. So my point is that we need to pay attention to this, that there's significant per dose cold chain expense. It's rational, but it's underappreciated. What are the risks? We look at these in two pillars, both from clinical, what happens if the cells don't work. So we administer a thermally sensitive dose that was exposed to a temp excursion. It doesn't work. Do we assay the cells before we administer? Not usually. Do we know if the patient didn't respond to that cell-based therapy because the cells didn't work? Maybe they were dead. That's a problem. That's one of the quality bars that we're trying to help this industry raise. And then look at the economic side. So scrapping a dose that couldn't be used, it arrived too late, it couldn't be administered during its sterility profile. This is the screenshot of the Biologistics Cold Chain SAS. It was recently awarded an IoT Asset Tracking Award, so we're proud of that. And Biologistics and the Evo SmartShipper really support these healthcare macro trends in terms of data management. To us, this is really all about data. So in our view, data loggers and individual data loggers that you take out of a package and stick into a computer later, those will be a thing of the past in the next few years. We want to be helping you assess data for every shipment, validating shipping lanes, validating a number of parameters on the health and quality of cells before they're administered, but definitely from a data visualization and aggregation standpoint afterwards. This is the family of Evo SmartShippers. It runs in 3 different operating modes. 2 to 8C with cold packs, CRT with different cold packs. These are very simply conditioned, and then cryo or -80 used with dry ice. I have a blow-up of what the Evo looks like that I'll show you. So on the app itself, really the sizzle here is that now users of the app can be recipients of alert messages on actionable data, and they can set up alerts to be pushed to the destination. So clinicians could understand the Evo and the shipment is an hour away, or it just arrived, or the lid was open, meaning it's in the facility, somebody opened it. Most importantly, that the shelf life is expiring. So imagine getting an alert that says there are 18 minutes left to infuse. That could be actionable, that could be meaningful. That's what the Evo looks like, very simple Packout. It's designed to eliminate Packout errors. You can only load it one way, integrated thermocouple, so you get true payload temperature because the thermocouple is right sitting against the actual biologic payload. The competition, it's foam coolers, beer coolers, if you can imagine that, or these really complicated vacuum insulated panel containers that you have to assemble each time, and hope you do it the right way. This is a big market for us as well. We think that of the overall biologistics market, for us, the slice of small form factor for cell therapy is perhaps $500 million. So the catalysts as we see them for us, it's all about creating value and then obviously capturing value. It's the media franchise we built, a built-in customer base to introduce biologistics to in the Evo SmartShipper, the IoT expansion, the growth of SaaS applications, data mining, data visualization for sure, and then really the increased regulatory scrutiny for cell therapies. We believe that over time, regulators and insurance payers will be much more interested and have a lot more scrutiny on the quality of shipping containers and data to make reimbursement decisions. Thanks very much. I think I did it just under the wire.